in the US, and he will talk about his research story about plant flower diversity. Dr. Yuan, please.
Thank you, Wenjia. Can you see my screen okay?
It's showing your, oh, yes, it works now.
Yeah, thank you. And I would like to also thank the organizers for this great opportunity to share some of our recent work, as well as my love for flowers. So I was initially trained as a plant taxonomist because the flower is usually the most informative organ to identify and classify plants. So I spent a lot of time looking at all sorts of flowers when I was a graduate student. What I was busy with classify and identify those plants. And just once in a while, I couldn't help to think that what makes this breathtaking beauty and diversity of flowers? Now, what are the genes regulating those remarkable pigmentation patterns and those elaborate floral forms that during the development of a single individual flower? And then how do the flowers change from species to species so much that during evolution of the species? So if you think about just the insanely large number of angiosperm species, which are 300,000 species, it is daunting to even just think about those questions. But when I started my postdoc career, I started realizing we don't have to study all of the 300,000 species to address those questions. And we can, in fact, carefully choose just one group of species, as long as they can represent a large space of the phenotypic diversity, and that can actually help us to understand the angiosperm diversity in general. And this species group I chose is called monkey flowers in the genus Mimulus. And one of the obvious reasons, as you can tell from this slide, is that they have a huge amount of diversity in flower color and form. And for example, among just the 4 closely related species we work on in the lab, those species diverged only about 5 million years ago, but you can tell that their morphology and pigmentation patterns are all very different. And in fact, those 4 species represent 3 distinct pollination syndromes. We have bee-pollinated species, a hummingbird-pollinated species, as well as a sulfur, just like Arabidopsis. And another reason that this group is really good is you can do genetic crosses between those species, and you can generate F2 populations like this that segregate with all of the floral traits we're interested in a very spectacular way. And that will help us to understand how the floral traits change from species to species. And when you work on natural variation like that, most of the floral trait variation between species will be polygenic. So it would be very useful if you can actually make new isogenic lines or introgression lines, as they're called in some other plant systems. The idea is that you can introgress just one locus at a time from one species to another, and then to look at what is the phenotypic consequence of this single locus. And for example, we can introgress some genes from this red flower species or hummingbird pollinated species to the sulfur. And this is the wild type sulfur. And you can change the color in many different ways by just changing one locus at a time. And these new isogenic lines can also help us then to look at the adaptive significance of those phenotypic changes. So if we're only changing one locus at a time, and we're only changing one phenotype, then what does that mean to the ecology of the species? So those new isogenic lines would provide the perfect material to address that question. And we can do then both common garden as well as field experiments in the natural habitat to observe what would the pollinator respond to the phenotypic changes. And in fact, about 20 years ago, there was a classic study by introgressing just one locus, affecting the yellow color, the yellow color variation between the 2 species. When you change this one single locus, you make this new isogenic line. Now the flower becomes yellow. And when you put that yellow new isogenic line together with the wild type in the natural habitat, this single locus change will decrease bumblebee visitation to the wild type about 7-fold, and it increases hummingbird pollination about 70-fold. So essentially, a single locus swap can produce a pollinator shift, which means that you basically generate a new species by changing a single locus. So finally, like I said, I'm interested in the diversity of what angiosperms. So there's another feature of this system is very interesting because you can actually generate a lot of just regular EMS mutants from one species, in this case, the bumblebee species. And those mutants are not just those odd, uninteresting ones. The phenotypes, in fact, they mimic some of those natural species that belong to a completely different plant families. So by studying those EMS mutants, supposedly we can then identify the genes and pathways that control the floral trait diversity in other species. So when I started our lab at the University of Connecticut, we decided we're just going to follow up on those two directions, research directions. One is to just look at using those mutants to look at the developmental genetics, the basic genes to discover new genes that regulates those understudied floral traits. Because a lot of those floral traits, they are in fact very prevalent among angiosperm species, but because they don't exist in the classic plant genetic model system of Arabidopsis, so people don't really know much about that basic genetic control. And then the other direction, of course, is on the evolutionary side. So we want to look at the molecular basis as well as the genomic basis that facilitate the changes, those rapid changes from one particular pollination syndrome to another among the species. And I'm going to very briefly mention just 3 floral traits and how I use the mutants to understand the basic developmental genetics. One is carotenoids, the yellow pigments. They're actually found in many flowers. But because, again, the Arabidopsis and Snapdragon, Petunia, those classic plant genetic model systems, they don't really produce carotenoids. So how the carotenoids are, when and where they're expressed, they're accumulated in the flower, is basically nothing was known before we started this whole study. And then the second trait is the floral tube. So more than 80,000 angiosperm species, you have those petals actually fused into a corolla tube. And those tubes are super important for, as you can tell, the hummingbirds, the hawk moth, for the special adaptation to different pollinators. But again, because Arabidopsis doesn't have a tube, so we don't really know much about what makes that tube. And then finally, you can see that some of those pigments are actually forming those very intricate spatial patterns. And we don't really know anything about, those spatial patterns, how those patterns are produced. Well, we shouldn't say anything. We know very little about the spatial patterning. So the first trait, the carotenoids, and the bee pollinator species, as you can tell, wild type, there's actually 2 yellow stripes on the lower petal. And that's actually the likely guide. So this yellow contrast with the pink tells the bee this is the way to come in. In fact, in the wild type, this is exactly how the bees actually go in. But in some mutants, when we get rid of the yellow, and the bee doesn't really know the direction anymore, they actually go into the flower like upside down. And we have many mutants in this bee pollinator species. For example, in those, I'm showing 2 of them called the reduced carotenoid pigmentation mutants. As you can tell, the yellow, like the guide, become much reduced. And by categorizing those mutants, we discovered 2 new regulators that controls, that basically are master regulators of the whole carotenoid biosynthetic pathway. And one of those genes is actually going to become important in the second half of the talk. And then the second floral trait we're focusing on is the corolla tube. So again, this is the wild type of bee pollinator species, and this is a back view of the corolla. You can see there's a very beautiful corolla tube. And again, the tube is important because they facilitate those very specialized pollinator adaptations. And we have a bunch of those corolla tube mutants. By analyzing the first 2 of them, we discovered that the key to this tube formation is really the synchronized growth between the petal primordial during the very early stage of flower development. You have the petal primordial, and then there's this interprimordial zone. So the synchronized growth between the base of the petal primordial and the interprimordial zone is really the key to the formation of the tube. Because the mutants, as you can tell, that the synchronized growth is gone, and now you don't have the tube. And then once we look at auxin signaling, it turns out that auxin signaling is very concentrated in this synchronized growth zone in the wild type, whereas in the mutant, it's very much reduced. And based on those analyses, we proposed this new conceptual model that highlighting the central role of this auxin signaling in the synchronized growth zone. And now we're analyzing both the upstream genetic networks as well as the downstream effector genes of auxin signaling and how they actually control the tube formation. And In addition, we're also doing live imaging. Well, this is an image of a oxygen reporter as well as a plasma membrane reporter, so we can actually track the cell growth. This is a 24-hour difference, and then in combination with mathematical modeling, and we can actually then simulate how the tube is growing during development. Now, the third trait I want to mention briefly is the pigmentation patterning. So in a lot of the flowers, see those spots or stripes. And people don't really know much of how those different patterns, those really intricate patterns are formed. And this problem, of course, it goes way beyond just the plants. Because in animals, you have a lot of those similar patterns. You go from a squid to bird fighters, from tropical fish skins to zebra stripes, right? So biologists, in fact, have been intrigued by those patterns for centuries. And the first theoretical model that proposed to explain the spontaneous formation of those spatial patterning from an initial uniform condition is actually by Alan Turing. And that's why this model, the so-called reaction diffusion model, is also called the Turing model. And he proposed that as long as you have two molecules, the activator and inhibitor, as long as they fulfill a set of requirements, you can actually then generate those heterogeneous spatial patterns out from an initial uniform condition. And I'm not going to go into the details of the parameters, etc. But it's fair to say that after many decades of searching, that people still don't really have good examples of what would constitute such an activator and inhibitor that actually fulfill what is the Turing model. And very recently, we found actually this pair. We published a study like describing the first pair of those proteins in the monkey flower system. And as you can tell, this is the wild type. You have the spots over there. And then when you lock out the activator, the spots are entirely gone. But then when you lock out the inhibitor, the spots got massively expanded into big patches. And we can, in fact, simulate, based on the mathematical model, and we can simulate the pigmentation pattern generation. This is like a condition that mimics the wild type. And then this is just a RNAi of the inhibitor. So we can basically artificially increase the degradation rate of the inhibitor a little bit. And this is the real plant phenotype. And you can tell corresponding to the simulation matches that very well. But of course, in this previous study, we are simulating things on just the arbitrary sort of geometry. I mean, no pattern looks really just like a square, right? So, and also we have entirely ignored the growth dynamics of the pattern because the pigmentation pattern is a part of the tissue growth. So right now we are incorporating both of those factors, and we are simulating the pattern formation process on the real geometry, like a real flower petal shape. And this is a dissected view of the whole corolla. So there are 5 petals, as well as a simulation of the growth, how the pattern are formed during the growth. And those simulations can actually guide us to design lab experiments to then even transgenically manipulate the dynamics of this reaction diffusion process to generate either larval patterns or just a lot of pattern variation. For example, I'm showing a few of those over here, and this is the wild type, and by just transgenically manipulate some of those parameters, you can actually generate many different types of pigment patterns. So those are 3 just very quick examples of the kind of floral traits we're studying that are before we didn't really know much about the basic genes and the development mechanisms control the growth of those organs or shapes. And then the other side of the lab is doing is, again, we're coming back to the original interest, which is the lateral variation. So we're looking at what are the genes, what are the genetic bases, the molecular bases that actually enable the phenotypic evolution among the species. I'm going to talk about just 2 quick examples, and both of them involve flower color and involve the difference between this sulfur as well, and this hummingbird pollinated species. So the hummingbird pollinated species is a dark red color because it has a combination of a high concentration of anthocyanins, which is on the upper layer of this tube, and carotenoids, which are yellow pigments in the lower layer. And the sulfur has a very pale, have a very little anthocyanins, and have low carotenoids at all. And then the first gene we map is the gene responsible for the anthocyanin variation. We will integrate the anthocyanin gene from the cardinalis from this red flower species into the sulfur, and you can make this new genic line. And now everything else is the same as the wild type, except now you have quite a bit of anthocyanins in the petal lobe. And when we first genetically map the gene, in this interval, there's a very obvious candidate gene. The R2R3 is known to regulate, to activate anthocyanins. So we thought, okay, this must be the right gene. But when we look at the two alleles between the two species, we realize that the transcript level, this gene is transcribed in both species about the same level. And that doesn't look like there's any amino acid sequence difference between the two alleles either that would make a difference in the function of the protein. So we were very puzzled by this for a long time, and then eventually we realized that the difference is actually not in the transcription of the gene or the protein function. It's actually in the 5' UTR region. So this is the four species I showed you along, and those are the sequence alignment. So this is the normal ATG start codon of all four species. In this sulfur, there's another ATG, just a few nucleotides upstream of the normal ATG start codon. And it turns out I didn't really know anything about this until we started digging into this. It turns out that those so-called upstream ATG or UATG are very prevalent in messenger RNA, in the 5' UTR region of messenger RNAs in both plants and animals, which I didn't know anything about before. And it turns out those UATGs are often play a repressive role in protein translation. So then we suspect, okay, this must be the cause of mutation. And we tested that by transforming the wild type allele from the sulfur into the sulfur background, and nothing changes, of course, because this allele doesn't really have a function. And then again, this is the real picture. And then when we just made we transformed the wild type with the same construct, except with one mutation. So we changed the UATG, the T here, into a G. And then you transform this into the wild type background, the color come back. So this shows that this definitely is the cause of mutation. And then subsequently, we made a reported constructs to show that yes, this mutation does affect the protein translation. So this is an example actually sort of really changed my views of how evolution occurs in a very fundamental way, because before that we always think that phenotypic variation is caused by either gene transcription difference or by protein function difference. And we very rarely, the molecular biologists might have thought about UTRs and other things, but for a lot of evolutionary biologists, I guess I can speak for myself. Now, we're rarely thinking about proteins. We're rarely thinking about protein translation. Just because, a transcript level difference, you can simply answer that by a qPCR or RNA-seq. And then protein coding sequence variation is very easy to identify. But a lot of times, the variation on the protein level, on the translational level, we don't really have a good way to answer that. So a lot of times we just ignore them. But I believe that those actually 5' UTR, those UATGs probably play a much more important role in generating natural variation than we used to think. And then the second example is about this yellow gene. Now, if you recall from what I told you before, there's this classic speciation gene, called yellow APA or YAP, that determines whether you have or not have carotenoids on the petal lobe. So when we introgress the gene from the hummingbird species into the sulfur again, and then now you have a little bit of carotenoids, so the petal become a little bit light orange. And when we genetically map that, now the first surprise is that this gene is actually very, very, very close to that gene responsible for the anthocyanin variation I just told you about. It's actually only one gene away. So essentially, these little fragments constitute a super gene. Now, even the gene between those two actually also play a role in flower coloration. So somehow you got this bunch of genes that all play an important role in flower color variation. They're clustered very closely together, forming a super gene. And as you can imagine, there's actually an advantage of that, because once you have an advantageous combination of alleles, you can generate flower color switches very quickly without the worry about, they are broken out by recombination. And the second surprise is that this YAP gene is actually not even a protein coding gene. It turns out it's actually a long coding, produce a long coding, a piece of long coding RNA, and then this long coding RNA locus, it derived from a source gene, a CYP450 source gene. And the third surprise is that one of the small RNAs actually is the critical small RNA and regulating the carotenoid pigmentation. But the source gene, the CYP450 source gene, had nothing to do with the flower coloration. So one of the small RNAs, in fact, the small RNAs produced by this stem loop structure, they actually form a phased pattern, so the phased RNAs. And one of these phased RNA actually targets RCP2, and that's one of the new regulators we discovered by characterizing those mutants earlier. And when you overexpress just this one little piece of small RNA alone into a genetic background that produces carotenoids, you can just turn off the carotenoids right away because this small RNA repress RCP2, and RCP2 is required for accumulating carotenoids. So that's the third surprise. The small RNA, it actually targets. So normally when I think about small RNA, it targets the source gene, right? The CYP450 gene. But in this case, it doesn't really target that gene. Instead, it targets a gene, an RCP2 gene, which had, evolutionary or phylogenetically speaking, had nothing to do with the source gene. So that's really another, the third surprise. And then finally, the fourth surprise is that the origin of this super locus is actually very recent. It's only originated about 5 million years ago. Most of those other monkey flower species don't even have that. So the upstream and downstream genes, that's very less collinearity in the genome. But this piece, this super gene locus, is not found in any of those other species, and it originated again in only this small clade of monkey flowers about 5 million years ago. And that's also very much against what I used to think about evolution, because if you ask any evolution biologist, we all think that phenotypic variation among species are usually caused by subtle expression change or protein coding or protein function change in those conserved developmental genes, developmental regulators. And this is so-called the taxon restricted or lineage specific genes. And we usually don't really think that they play an important role in species evolution, but it turns out that those genes are probably do play a much more important role than we used to think. In fact, this super gene locus, this taxon specific super gene locus, played a very important role in not just flower coloration, but also speciation and reproductive isolation, as well as the pollinator shifts. So those are the really four big surprises from this one single locus. And then finally, there's a personal sort of a note that this super locus is very satisfying just personally to myself. It's because we actually spend a lot of time characterizing those mutants, the EMS mutants, like RCP2, etc. And if we didn't do that, even if we have genetically mapped this locus, we would still have no idea how does this small RNA locus actually function without previously characterizing the RCP2. So this is really a combination of those two major directions of research. They kind of converge together and help us understand phenotypic variation. And finally, we're not only working on those flower color variation, we're also working on flower size and the petal shape, as well as those reproductive organ like pistil length, etc. And hopefully, in another 10 years or so, we will be able to understand much better, much more about the genetic changes like causing phenotypic variation, like causing this tremendous diversity of flowers. I would like to end by thanking my labs and funding agencies. Thank you.
Thank you, Dr. Yuan. Very interesting talk. So now is the Q&A session. Any questions from our committee members?
Yeah, I'll ask one then. Always nice to hear that 5 million years ago is recent. So, but what I found interesting from my background is that you mentioned Turing patterns in this pigmentation patterning. And so you suggested two MIP factors that would act there as activator and inhibitor. But in the original implementation of these Turing models with the spatial aspect, there is kind of this requirement that the inhibitor is mobile and is faster than the activator. So are you saying that those MIP factors are actually cell-to-cell and mobile? Has that been shown somehow?
Yes, so we actually have shown in the previous paper. I didn't show the specific data in this talk, but yes, especially the small inhibitors, actually very small proteins, only about 80 amino acids long. They definitely diffuse from cell to cell.
Okay. Cool. Thank you.
And in fact, one of those transgenic manipulation, the picture I show is actually is a color synthesis transgenic line. So we're artificially producing more color synthesis to block the plasma desmata. So that way you can actually change the diffusion rate of the proteins, and then that changes the pigmentation patterns.
Okay, thank you.
Yeah, maybe another question, but I think I didn't read the paper regarding the corolla tube formation, but of course the phenotype. So you come to the auxin with the DR5, so which is very similar to what early experiments people have seen when they treated like Arabidopsis spin mutants with auxin, NAA and NPA treatments. This lanolin. I guess you did kind of experiments in this species to put locally NAA and to try to interfere with the corolla tube formation.
Yeah, very good question. So yes, we did actually treat them with MPA, and MPA would totally disrupt the corolla tube formation. But we didn't really do the localized application because that was really hard for this species. We did the sort of a brutal way, just we spray the MPA onto the young fluorobuds and just infiltrate the heck out of it. so that way it's not a very precise application, but it did result I think it's about 20% of the flowers actually coming out from that application turns out to have low corolla, has disrupted corolla tubes.
Yeah, I see. So it's probably also has to do with the distribution of auxin in these meristems that have to be localized in some way to create this corolla. And do you have? I guess you can also go for CRISPR-Cas approaches in these plants or not?
We can, and we actually have a lot of CRISPR lines I just didn't show, and again we are working on both the upstream and downstream of this. So again, you're totally right. The key to the formation of the tube is the precise localization of the auxin signaling. So there's actually a lot of leaf polarity genes. It turns out they actually affect the localization of the auxin in the synchronized growth zone. And then obviously also the effector genes, like what's downstream of the auxin signaling, right? What is really repressing the cell growth or induce the cell growth, etc. And not surprisingly, one of the genes is the Arabidopsis COX2 ortholog. That actually play, if you overexpress that COX2, you also don't produce the tube.
Okay, I see. Okay, thank you very much.
Thank you.
Hi, your next talk. So in the last part of your talk, you talk about a very interesting